light dependent reactions. If you've never seen that abbreviation before, RXN, it just means reaction. It saves you a couple letters. It's kind of nice. All right, so let's talk first about what the light-dependent reactions are going to do. Obviously, we know they're dependent on light, so they have to have light. This is the energy capture phase of photosynthesis. We know that photosynthesis is going to use light, and so we're going to capture energy from light, and we're going to produce two molecules. Okay, molecule number one is ATP, so it produces ATP, and molecule number two is that high energy electron carrier that's used in photosynthesis. It's called NADPH. Again, you can remember that NADPH is the one that is used in photosynthesis because it's got the P in it. Even though that's not really what the P means, you can think that it is. Okay? So, where does the energy come from then? Photosynthesis. Sunlight, right? Okay, so how do we get energy from sunlight? Energy from sunlight is captured by molecules called pigments. Okay, and you guys have heard about pigments before, probably. They are the things that give stuff color, right? So you could say like, oh, if I'm trying to paint something, I'm going to need to put in these pigments to get the colors that I want, or something like that. Okay. The specific pigments that are probably the most important for plants are chlorophylls. Okay, there's two chlorophylls, chlorophyll A and chlorophyll B. There are some other ones too. There's ones called xanthophylls, and there's ones that are called carotenoids. They are all going to absorb different wavelengths of light. And it happens to be that chlorophyll A and chlorophyll B are really good at absorbing reddish light. Chlorophyll A is best around 665 nanometers wavelength, and chlorophyll B is best around 640 nanometers wavelength of light. And I want to talk for a quick second about what this means. We know that light is carried on this thing called the photon, right? But light is more than light. Light is just one little piece in this thing called the electromagnetic spectrum. So this is the electromagnetic spectrum. And the electromagnetic spectrum is, is all the waves that are produced when things travel at the speed of light. Okay, they're produced by the photon, carried by the photon, and they are all things that are traveling at the speed of light. Okay, now since you're traveling at the speed of light, there's two properties of each wave that tend to be important in this case. Okay, the first property is the wavelength, which if you draw a wave like this, it's the distance in between the peaks and the troughs. Right, so peak to peak or trough to trough, that gives you the wavelength of a wave. Okay. The second thing is the frequency, or how fast it's moving, how many times that wave goes up and down per second. Okay. So in the case of the electromagnetic spectrum, since they're all traveling at the speed of light, there has to be an inverse relationship, meaning if you have a really long wavelength, it moves slower, and if you have a really short wavelength, it moves faster. Right. So low wavelength, high frequency; high wavelength, low frequency. Right. Okay. So down at the very bottom are your lowest energy things. Those are radio waves. Then you've got the next group, which is microwaves. Okay, then you got the next group, which is called infrared light. Then you got a very small sliver here. We'll talk about it in a second. Then above that sliver, you've got ultraviolet light. Then above that, you've got X-rays. And above that, you've got gamma rays. Okay, so gamma rays are the highest energy because they have the lowest wavelength and the highest frequency. Radio waves are the lowest energy because they have the highest wavelength and the lowest frequency. And this is good because there's radio waves all around us right now and they're not killing us. And I know that there's radio waves around us because if I had a radio, I could hold it up like this and turn it to 97.1 or something and I could get music, right? Another radio wave is wireless internet. So like if you get Wi-Fi, that runs on 2.4 gigahertz, right? And that's telling you the frequency of it. And so that's in the radio wave spectrum, okay? The next one is microwaves. Microwaves are what your cell phone runs off of, and those are important. And so we know that cell phones don't kill us, hopefully. And they don't because it's a pretty low energy wave. And so we don't really have to worry about that. The next one is infrared light. If you like shine your remote at the TV and you press a button, infrared light comes out of it and it tells the cable box to change the channel or something like that. Okay, that's how that works. And then the next little sliver there, we didn't write anything for because that's probably the kind that's the most important for you and me. Okay, and it's not necessarily any more important than the rest of them, except for the fact that this is the kind that we can detect in our bodies. So this is Roy G. Biv, which are the colors of the rainbow: red, orange, yellow, green, blue, indigo, and violet. And this is all of the visible light. Okay, that's not to say that visible light is more important than any other type of light. It's just the type that's more important to us because we can see it. Okay. So plants need infrared light, they need visible light, and they need ultraviolet light. Okay, that's why if you try to just take like a normal incandescent light bulb and shine it on a plant, it's not going to grow super well. You need like a grow bulb, and a grow bulb is going to have a higher range of UV spectrum in it that will allow it to get more energy. Okay, so we talked specifically about chlorophyll A and chlorophyll B, and so if you know the range here, red is about 700 nanometers. And violet is about 400 nanometers. And so the band that chlorophyll A and chlorophyll B are going to take up is right about the end of red and the beginning of orange. Okay, so this is the most important light for plants. And we know this because this is the light that is going to get absorbed by those pigments. And the pigments are the things that are going to actually capture that light from, or capture the energy from that light. Okay? That means that when you look at a plant, you would expect it to be red? No. When you look at plants, they're not red, they're green, right? So why are they green? Because green is here. It is not part of the stuff that's being absorbed. So the only thing that we see are colors that hit the plant, and the plant doesn't need them, so they get reflected away as essentially waste, you can think of them. So when I look at this tree here, it's green because the sunlight is hitting it, and it's absorbing all the red light, and it's just shining the green light back at my eyes. Right? What's that? Because they're being absorbed, and because a lot of them, when combined, make a greenish tint. But when you look at all the things that are being absorbed, the thing that is absorbed the least is green. Okay? Now, if it absorbed all the light, if plants were black, they wouldn't work out because they would just die. They would get cooked by the sun and they would die because that would mean they were absorbing all the light, and that would be too much thermal energy, too much solar energy, and they would just get cooked, right? So they have to get rid of some of the light in order to not get cooked. All right? So let's talk about how this is used by the plant. Okay, so we're going to draw a pretty detailed here thylakoid. The black stuff with squigglies like that, that's going to be plasma membrane. And we've got a little green area that's called an antenna complex. And in the antenna complex, there's going to be a bunch of pigments, a bunch of chlorophyll molecules. And then in the center, a really big complex that's called the primary electron acceptor. Okay? Then you've got a little bit more plasma membrane. And then you got some embedded proteins in here. One of them is just integral, and one of them is transmembrane. Okay, then you've got a little bit more plasma membrane. Then you've got another one of these things, these antenna complexes. And in the center, a big primary electron acceptor. And a little bit more plasma membrane. And another little intermediate protein here. And then a bigger enzyme on the end. And then this is going to curve around. Because what we're looking at here is the thylakoid, and we know that the thylakoid, in order to function properly, needs a thylakoid space, or a space in the middle. Okay, and then on the bottom, we're going to draw one more protein enzyme. And now we've got a decent thylakoid. This is all plasma membrane in here. So we'll just quickly put in a couple of phospholipid tails so that we know that we're talking about plasma membrane. Uh-oh. What just happened? Come back to me. Not too bad. Okay. Something like this. All right. So let's label some stuff. This area here is an area that's called photosystem 2. Okay, and we are just going to abbreviate it up here. We'll write it down below. This is PS2. Okay, this area right here is called PS1. And you might immediately say, well, that's stupid. Why does PS1 come after PS2? And the answer to that is because it's stupid. Unfortunately, PS1, Photosystem 1, was discovered before Photosystem 2. And so they just called it Photosystem. And then they found another one. They're like, well, look, there's another photosystem here doing different stuff. And they're like, what do we call it? And they're like, I don't know, call it Photosystem 2. And they're like, but what if it's the one that's actually working first in this process? Like, I don't know. We'll figure that out later. So they called it Photosystem 2, and then later on they were like, oh man, that is the thing that's going first. And so they were like, what do we do now? And they're like, well, we've already taught everybody that that's Photosystem 2, so we'll just keep it. And they just kept it. So Photosystem 2 actually goes first here, and then Photosystem 1 comes afterwards. Okay? This thing here is an H+ ion pump. This thing here is an enzyme called NADP+ reductase. And this guy down here is an enzyme called ATP synthase, which you guys know is a channel protein, so we should draw the channel in it. Like so. All right, you guys ready to learn about the process of the light reactions? Step 1. Energy from sunlight is absorbed in photosystem 2 by pigments. Okay? You guys remember doing the flame test lab in chemistry? The one where you take the popsicle sticks and you dip them into this stuff and then you burn them and then you burn different colors? Everybody do that? Anybody do that? No? You should have done it. If you didn't do it, you should have. Anyways, it happens. And the reason why it happens is because when you put those things into a fire, the electrons jump to a higher energy level, and then they fall back down. And when they fall back down, they release a photon, and the photon's of a specific spectrum, and they call that the atomic emission spectrum. That stuff's not all that important. What you need to know is when you add energy to an electron, it jumps to a higher energy level. Okay? Now, you can do it with a flame, or you can do it with sunlight. So as sunlight goes in here, the electrons that are in these pigments, they jump to a higher energy level. And if it was a flame test lab, they would fall back down and emit a photon of a certain energy, a certain wavelength of light. But this isn't a flame test lab. This is a controlled environment here. And so the electrons that jump to higher energy levels are immediately passed into the thing in the center here, which is called the primary electron acceptor. So we can say that high energy electrons are passed into the primary electron acceptor. Okay? This creates an electron deficit in the pigments. They have less electrons now than they should. And so this gives them an ability then to take electrons from other things. When you have something that has an electron deficit, it becomes really good at taking electrons, which means that it's going to be able to cause a reaction to happen, right? So we can say that the reaction that happens here is that the energy then from the electrons moving to the primary electron acceptor is used to split water. Energy in PS2 is used to split water. Into H plus ions and O2. This process is called photolysis. Okay? Which actually makes a lot of sense. What does photo mean? Light. What does lysis mean? To break down. So we're going to break down water using light. Right? Photolysis. Yes? Here's what photolysis looks like. 2H2O yields 4 H plus ions plus O2. And we said that the energy here in photosystem 2 that was used to do this was caused by an electron deficit. And so what had to be stolen away were electrons. If you have 4 H plus ions, there's also 4 electrons in here that are broken free. Okay? And those 4 electrons then go and replenish the electrons in photosystem 2 that were passed on to the primary electron acceptor. Okay? Following so far? We split up some water. So we had water in here. 2 H2Os. They got split into O2, 4 electrons, and 4 H plus ions. The O2 is a small nonpolar molecule that's waste, so what happens? Oh geez, my drawing got all jacked up. Why did that happen to me? Okay. Small nonpolar. I bet they're way over here. Yes, they are. I don't know why that happens sometimes. All right. Small nonpolar molecules, what can they do? Diffuse out. So this is waste. So the O2 is just going to diffuse out of the membrane. The 4 electrons, they're going to replenish the electrons that were in photosystem 2. And we're going to talk about the H plus ions later. Okay? Step 3. High energy electrons are used to pump H plus ions into the thylakoid space. Okay, so you've got high energy electrons here. This is going to be a high energy electron we're going to show. It's going to get passed over here to this. That's going to lower the energy slightly so that it's the correct amount of energy now to pass into the pump. Okay, and then the pump is going to use all the energy. The pump uses the energy, and it pumps an H plus ion across into the thylakoid space. So now we got a low energy electron. Boo, low energy electrons. So they're going to get passed back into photosystem 2. So we'll say the low energy electron ends up in photosystem 2. It is passed to, or sorry, photosystem 1 rather. See, even I mess it up sometimes. Alright, so it's passed to photosystem 1, which is actually the second photosystem. Okay, step 4. PS1 re-energizes the electrons using energy from sunlight. So we got energy from sunlight coming in. It bumps this electron's energy back up. And then step 5 is it gets passed over into this intermediate molecule and then into NADP plus reductase. Okay, that's step 5. High energy electrons get passed into NADP plus reductase. Okay, yeah.
What are
The green things? These? They're called antenna complexes. That's where the actual pigments are. So all the chlorophyll, the stuff that's actually absorbing the sunlight and raising the electron's energy, they're in the green parts. And then the big green parts in the middle, those are the primary electron acceptors that actually take those high energy electrons in and pass them along.
Okay, so that's what's like responsible for passing it.
Yep. Okay. So, if you had to guess what NADP plus reductase does, what would you guess? Reduces NADP plus, right? And when we reduce something, we add electrons to it. So we're going to take NADP plus 2 electrons, and that's going to give us NADPH. And I know that's confusing, right? Because we didn't show that it was gaining a proton. But because this is in an aqueous solution, there's always protons around. And so whenever you have something that's going to gain a negative charge, it's never going to stay negative because a hydrogen ion is just going to smack onto it right away. Right? So it's going to become NADPH when it gains 2 electrons. Okay? Make sense so far? Lose anybody yet? So now we have taken these high energy electrons and we can put them on NADPH. Draw some squigglies around NADPH so we know there's a final product here. And then we also want to show that this has 2 electrons on it, so I'm going to draw 2 big electrons on it like that. NADPH picks up 2 high energy electrons, and then it can use those high energy electrons later on in the Calvin cycle. Okay, so we've created one of the things. We created NADPH, but we said we were creating 2 things. We were going to create ATP and NADPH. So we've got to find a way to make ATP. What's probably going to be involved in the production of ATP? ATP synthase. What does ATP synthase need? H plus ions. Did we make H plus ions? Did we put them inside a membrane? We sure did. So now all we have to do is take all these H plus ions and let them flow out through ATP synthase. And that creates ATP. We did it. We made both the things that we needed to make. So that's step 6. H plus ions flow from the thylakoid space into the stroma through ATP synthase to create ATP. Easy enough? I mean, really, when you think about it, like, it's a very complex process. I'm not going to lie to you about that. But there's only like 6 things that happen. 6 things isn't that many. It takes more numbers to dial a phone number, although you guys don't really dial phone numbers anymore, do you? Just search them on the internet and then press the button. All right. So let's watch it in action. Here we go. Step 1, we're going to split water. I know that's not the first step on there, but we're going to split water. Okay, so H2O comes in, the electron deficit causes it to split, your O2 goes out as waste, your electrons go into the antenna complex, and your H plus ions, the positive things in here, they stay in the thylakoid space, right? Split water, electrons go into the antenna complex, hydrogen ions end up in the thylakoid space, and the O2 diffuses out. Is that what we put? Yep. Is that what we put? We got that part? All right. Step 2, we're going to use the high energy electrons from photosystem 2 here, which is this part, to pump protons, or H plus ions, into the thylakoid space. As that gets reduced, it pumps a proton in. And then we have a low energy electron after that. The low energy electron then goes into photosystem 1. Right? Photosystem 1. And then photosystem 1 will take that low energy electron and re-energize it and then pass it over into NADP plus reductase. It only showed 1 electron there, but it's actually 2. It's just an error in the animation. Okay, and the last thing, all those H plus ions that we pumped into the thylakoid space, those are going to make ATP by flowing through ATP synthase, which the energy is then used to combine an ADP with a phosphate to create ATP. Questions on that so far? All right, there's one other thing that we got to talk about. I was hoping you wouldn't be confused yet, because this part. All right, so here's some stuff. Here's a thylakoid. This is photosystem 2. Here's the proton pump. Here's photosystem 1. Here's NADP reductase. So this is the pump. For H plus ions. This is the NADP plus reductase. And then down here at the bottom, we can put an ATP synthase. Okay, so what would normally happen is you would have an electron down here that's created through the splitting of water. It gets bumped up in energy by photosystem 2. This is PS2. This is PS1. Okay, it gets sent over to the H plus ion pump. The H plus ion pump takes its energy. So now it's a low energy electron. That gets passed over to photosystem 1. Photosystem 1 bumps its energy back up. And it gets sent into NADP plus reductase. This happens twice to make a single NADP plus. Everybody agree? That's what we just talked about? Okay. So, in this, we can all, I think, agree that this has a beginning. This is the beginning, right? And it has an end. This is the end, right? So it's just a path that it's going on. So we'd say this is going to travel along this path like this. Doop, 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 doop. Down here, up here. The way that we would describe this is called non-cyclic electron flow. Non-cyclic electron flow. Does everybody see that non-cyclic electron flow is not a cycle? It's non-cyclic, right? Yes? Non-cyclic electron flow at the end is going to produce NADPH. And it's also going to pump H plus ions in. And those H plus ions are going to flow through ATP synthase to create ATP. Yeah? Everybody agree? Okay, so this creates ATP. And it creates NADPH. The problem is that NADPH and ATP are not used in the same ratio in the Calvin cycle. They're not a 1-to-1 ratio. In fact, we're going to use a lot more ATP in the Calvin cycle than we use NADPH. This means that in any given reaction, ATP is going to be the limiting reactant, or the one that runs out first. And we're going to be left with an excess of NADPH. Okay? So we got to find a way then to make more ATP without making more NADPH. Okay? So, in the process here of cyclic electron flow, we end up with 2 electrons that end up in this enzyme here. Okay? So you can imagine this is like an enzyme that has an active site where the electrons fit in. Okay? And there are 2 electrons in there. If there's 2 electrons in there, the thing that's actually going to go and pick up the electrons is the molecule NADP+, right? So an NADP+ would normally come over and attach to the other spot in there, right? And that would make an NADPH, right? You guys agree? Have I lost anybody about this? NADP+ plus 2 electrons gives us NADPH. Yeah? Okay. What if there's no NADP+? What's that? Wait till there is one. So this electron here, these 2 electrons, they just chill out in that enzyme until there's an NADP+ to pick them up. Well, what if photosystem 2 makes more high energy electrons while that's happening? Or photosystem 1 rather, makes more high energy electrons while that's happening. It's got to go the other way. So instead of sending it to the NADP+ reductase, that spot is blocked. And so it energizes electrons, but instead of sending them over, it sends them back to the pump. The pump uses them to pump H plus ions, and then it sends it back to photosystem 1 again. So that's this cycle here.
How does it send them back to photosystem 1?
How does it send them back to photosystem 1 if what? Yeah, this is photosystem 1 right here. Yeah. So instead of going this way, it has to just send them this way. And so it sends them in a cycle. Right? So that's called cyclic electron flow. Because it's a cycle. Cyclic electron flow pumps H plus ions, right? And so what does cyclic electron flow create? ATP only. So in order to get the correct ratio of ATP and NADPH, we have to have the correct ratio of cyclic flow and non-cyclic flow. Does going through cyclic flow use more water? Cyclic flow? Where's the water split? Back here. Does doing this use any more water? No, so it just creates ATP for free. No reactants are used here. We're just using the same electron over and over again with sunlight to create ATP. Yeah?
So every time it goes in that loop, it pumps out more H plus?
Pumps in more H plus, yeah. You guys see how that works? Anybody have any questions on it? All right, that's it.